Okay, so I guess we're struggling a little bit on incomplete co and sex-linked crosses. So I'm going to try and help you out as best I can, and hopefully this will make more sense. So first of all, when it comes to incomplete dominance, we've got two alleles being inherited, but when one of the alleles is inherited by itself, it's not quite as strong as standard dominant versus recessive traits go. So if you remember that when we crossed and this is for purple flower color, that when we crossed a plant that was heterozygous for purple flower colors against another one that was also heterozygous, that these three of the offspring are going to be purple, but exactly the same shade of purple. This one is going to be white. Okay. For incomplete dominance, we do the same cross where we take a red plant, for example. I'm going to say big R, big R. In order for it to be red in this case, both alleles must be identical. And let's say that we cross that plant against one that is also white. Well, what makes incomplete dominance different is that these offspring are actually not red because not enough red pigment is being manufactured. So as a result, these plants are pink. Okay, so just a lighter shade of red because not enough of the pigment is being made. So if we then take the two pink offspring and cross them, this first offspring right here is actually red because it has both alleles that are coding for red pigment. Then we have offspring that are pink because they're not making as much of that pigment. And then we have offspring that are white. So what makes these crosses a little different is that we're getting a third phenotype. So in regular crosses and regular Mendelian genetics, we get those either or. So in this case, either it's purple and all the same shade of purple, whether it's homozygous or heterozygous, or they're white. With incomplete dominance, I'm no longer getting the heterozygote that looks like the homozygote. It now has its own phenotype. So when we look at phenotypic ratios, it's now actually 1 to 2 to 1. Because even though I have one capital R in this box, they're not red, they're pink. And it's not the same color as the red flower. So for example, let's say you breed a red carnation against a white carnation. Well, you don't get red carnations in this case, you get pink ones. So we're getting an entirely different phenotype here. Now, when we look at codominance, codominance is similar to incomplete dominance in that we get a third phenotype. So the cross is the same cross. Okay, so if we cross, say, two heterozygotes, okay, we still get the combination of big R, big R. We get big R, and little r, little r. So with codominant alleles, what ends up happening is that these plants would be red because they are homozygous for both alleles, and both alleles are making pigment. Then our heterozygotes would actually be red and white. So they would be expressing those pigments at exactly the same time right next to each other. Okay, so for example, if you've ever seen those pansies, you guys will notice that they kind of have the purple exterior but the white interior. That's codominant. If you take a look at tigers, for example, that have both the orange and black fur, or the white and black fur, That is also codominant. Okay. And then the offspring here, the little r bucks, these guys are just white as always. Okay. So the ratios when it comes to incomplete and codominance, the phenotypic ratio and the genotypic ratio will always be identical. So it would be